complex systems in biology, there's very little stuff that you can do analytically. So, because it's a many particle system, very complex interactions, you have to do some numerics, but then you can try to understand the numerics with some simple models. So it's both. Okay, but now, so what is chemical equilibrium? Why are we interested in it? Why is it important? You can look at the slide if you want, if it helps you.
I can see the energy, maybe the energy of exchange of, in the case of two particles, if you have some kind of interaction or any process between the two.
Yes.
The energy generated should be
Okay, so I have let's say a particle that can exist in two states. Okay. This is the simplest system to think about. A particle, it can exist in two energy states, and it has a certain probability of being in one energy versus another. Right? The equilibrium constant tells me what the relative concentration of being in this species is versus this species, with this state versus that state. So in physics, we know that this is related to a Boltzmann factor. Right? What the chemists and the experimentalists like to think about is ratios of concentrations. But they're the same thing. Okay? It's not the energy itself that matters. It's the probability of finding a particular energy. And the probability is e to the minus beta of that energy. So I'm going to go through what I talked about last time a bit better, because you're missing a step on where the partition function comes in. Okay, so just hold on a second. So so just some definitions first. So that's the equilibrium constant. That's what is known as the equilibrium constant. The inverse of it is the dissociation constant. Okay, so you've seen equilibrium constants from statistical mechanics already. Okay, it's just not been called that. So I have a particle. It can interconvert between two states A and B. Okay, and I want to know what the total partition function of that single molecule is. Well, if I want to do that, I just need to integrate or sum over e to the minus beta Ej. That can be separated into contributions coming from energies in state A versus those in state B. Okay, so the total partition function of my molecule converting between these two states is QA plus QB. Okay, that's the total partition function. Okay, that's for one particle, one molecule. Call it whatever you want. Now I have an ensemble of these n particles or n molecules. Okay, so the total partition function using the Boltzmann approximation is this binomial type expansion. Okay? Okay, so now we're getting closer to getting to the question of the concentrations. So the probability of finding Na molecules and Nb molecules will be given by a ratio of some constrained partition function divided by the total partition function. This is just the probability. Okay, so it's easier to work with the log natural of this. And if I want to know what the most probable value of NA is, then I just take the derivative of that, set it to 0. And when I do that, okay, and you simplify, what you see is you get the ratio of the concentration or the number of particles NA versus NB is just the ratio of the partition functions. And the ratio of these partition functions is e to the minus beta Helmholtz free energy. Okay, so the concentration, ratio partition functions, free energy, they're all connected. And that's why being able to measure, being able to compare to an equilibrium constant in an experiment is very, very useful. Okay, because you can, it links you back to the ratio of the concentrations, it links you back to the ratio of partition functions, and therefore the free energy difference between the two states, or the two populations. Okay? Okay. Fine. So this is just a review. We're now going to, in class, tackle this problem of not a single molecule, but a protein binding to a ligand, so another molecule. And this is a very standard problem in biology. So I have a ligand and a receptor, and there's an equilibrium between the ligand and the receptor being far away from each other, and then they bind to form a complex. This is a very standard thing in biology. So there's this equilibrium between L plus R and the complex. Okay, so this is how the chemists or the experimentalists like to write this. So this is the dissociation constant. So it's the ratio of the product of this concentration, that concentration divided by this. Now, what you can do is you can write out the probability of the ligand, the receptor site being bound by a ligand, and that's simply the ratio of this concentration to the ratio to the sum of this plus the number of receptors in solution. Okay, that's, yeah, please.
In this things, what is the general unit of concentration? Mole per liter or kg per liter?
It's usually in mole per volume. Mole per volume. So you'll see it in written as capital M, molar. And depending on the magnitude of that, that will tell you how strong the binding constant is. Okay, so this is something. Okay, if you use that definition of Kd, you can write P bound as this. This is something that an experimentalist can measure. Now, if you set Kd equals to L, you get P bound equals a half. What does this mean? This means that the dissociation constant, so the physical interpretation of dissociation constant, is the concentration at which the receptor has a 50% chance of being bound by a ligand. This is what the dissociation constant means. Okay, so I'm just giving, this may seem a bit esoteric at this moment, but I'm just giving you a perspective of how the experimentalist thinks of this problem. Now we're going to go down to a lattice model. We're going down to a lattice model, and we're going to try and see whether we can come up with an expression that looks like the following. Okay, so the question is, I have a receptor, a protein, I have ligands in the solution, and I want to know what is the probability that the ligand binds to the protein. That's the question. Okay, so, yeah.
The stuff you showed, is it a chemical reaction or?
It can be.
It's like a lock and key hypothesis. So
it's a what, sorry?
Like a lock and key.
This is any equilibrium you want to think of. So it can be a chemical reaction, or it can just be two things, lock and key. It can be whatever you want it to be. So it's like, for example, you take hydrochloric acid and you put it in water.
Because the last statement you made that
Yes.
50% chance of the receptor being occupied.
Yeah.
So for me, it looks like there is a site that the other one has to fit into, and perhaps if for example let me see the right side.
Yeah.
You have like just some empty states, so many empty states, and then the left side. And then we have two, so we have to choose among which one to take.
So you're talking about a more complicated problem. So in this language, so this expression here doesn't allow you to say anything about the number of sites where the ligand it's a thermodynamic equilibrium of, let's say, reactants and products and their concentrations. That's all. You don't have a window into the type of thing that you're thinking about. But it's an important question. Okay, and it can affect this. Okay, it can, but we're not going there yet. Okay, so let me present to you what the model is. Okay, so this is what you call your computer game, but it's not.
And that is really the lock and key I'm talking about.
What's the lock and what's the key?
See the last picture.
Yes.
See the guy fit exactly inside
Yeah.
the hole. So
Yeah.
there is a term in biology called the lock and key hypothesis.
Yes.
Where you have some sites.
Yeah.
They are like the lock of the door.
That's right.
And then the key.
That's
and
right.
the fit that
That's right.
I mean, but you have so many enzymes, so they look for a particular site that is attractive for them. The fitting site.
Yes. Okay. The lock and key hypothesis. Well, it's a hypothesis, and it's been shown not to be true. It's completely incorrect.
I watched one and they use it's true because they use the hypothesis to explain some kind of biological process.
That's true. People make all sorts of hypotheses, and it's true that in biology people used to invoke this hypothesis.
Yeah.
And I'm not saying it's not entirely incorrect, but it's very simplistic. So it neglects many things that we can talk about.
Okay.
But so you're right. Okay. If over here for this picture, okay, let me explain the picture then see if it clarifies what you have in your head. So I have a lattice. I have many ligands in the lattice. I have a protein, and the ligand has to stick inside here in order for the function to happen. Okay, it doesn't matter what the function is. So there are many sites where the ligands can be on the lattice. They have a certain energy associated with their interaction of the ligand with the solvent. Okay, I'll explain that more later. And then when it sits into the site, it has some interaction energy with the receptor, a binding energy. Okay, so we want to see if we can ask the question and answer, what is the probability of finding a ligand in the receptor site? That's the question. Okay? So, what is the probability of the receptor site hosting a ligand? Okay, so that's the question. I have omega lattice sites. I have L ligands. I have E sol. This is the interaction energy of the ligand with the solvent. So when the ligand is out here, each ligand has an interaction energy of E sol. Okay? And there's E bind. So when the ligand interacts with the receptor, and only one ligand can bind to the receptor site, I get an energy of E bind. So these are the parameters. Okay, and so we want to know what is the probability of the receptor site hosting a ligand. Okay, so how do we do it?
So there can be only one receptor in the ligand, and
only one ligand in this. Yeah.
So if we let's suppose for some time that we don't have 4 and 5.
Let's say we don't have 4. Perfect.
So if we don't have 4 and 5, it's a random choice. So
okay, good.
So one picture would be we picture number 4, and there are, let's say, a small n number of ligand. And so now one picture in the question we are interested in just picture number 4, microstate 4.
Well, we want to find the weight of finding microstate 4.
Okay, let's okay, let's put weight later on.
Okay, so you want to start without the weight.
Let's
Okay,
put
great.
weight later on.
Okay.
Because we know the weight of a picture.
Yeah.
We know weight of a picture we are interested in. We know
Yes.
the weight of the picture we are not interested in.
Great.
So the weight of we are only interested in picture number 4.
Okay.
Because that is when the binding happens.
Yes.
So if I don't know, can we paint all the ligands differently in different color?
Why would this help you?
Because otherwise all the pictures would be same, kind of like I can replicate the same picture by exchanging one ligand that is bound with another one, and it will be a same picture. If I paint differently.
So, well, okay. So, so here the ligands are indistinguishable. Okay, they're indistinguishable. But along the lines of thought, this is why we need to think about putting the weights.
But isn't the weight same for all the?
The weight is the same for all the ligands when they're in the bath.
Okay, so let's do this.
Yeah.
So let's have this picture.
Yeah.
So one ligand is gone.
Yes.
So n-1 ligand is left.
Perfect.
So they can choose L lattice site in omega C n-1. Omega C L-1.
Excellent. Okay. Good. So now I want all of you to start. So you're thinking in the right direction. So two things. One is I need to define the partition function of my system. First of all, the partition function will be the sum of all microstates where the ligand is not in the receptor site, plus the sum of all microstates where the ligand is in the receptor site. So I have what you described is the combinatorial part of it, but then I need to put in the Boltzmann weight.
Yeah.
Okay? So why don't you, so first, does everyone understand the question? Okay, so first thing, let's assume there's no ligand in the receptor. Okay? How many different ways are there to put L ligands on omega sites? That's it. The first thing you should ask yourself. And write it down for me.
Please repeat the question.
Well, no. So this is the question.
You want the probability.
Yeah. So I want the probability of finding a ligand in the receptor site.
And one more thing, like even if it is out of the receptor, no matter where it is, the energy is so.
Yes, that's the assumption.
omega is bigger than the number of ligands.
Definitely. Yes, that's a good point. Thank you for saying it. Because actually, in order to simplify things, the number of lattice sites is much greater than the number of ligands. You don't need this now, but
Excuse me.
yeah.
So what I'm doing is find Z.
Yes,
So
perfect.
in this case, I divided into 2 parts because we
Yeah.
have to consider all the things. So here it is all the pictures in which it is out of the
So all the ligands are in solution.
again solution.
Okay.
This is the weight which I just calculate like omega CL.
Well, okay, so this is the multiplicity you mean, and this is the product is the weight. And what do you mean by because this
Well,
itself is
it
not,
has to be here. I think.
yeah, exactly,
That's
exactly.
what
Okay, good.
And here, one ligand is inside. This is the weight. And
Yes.
it should be here. Okay, that's where now it is the because one is inside. So
Yeah.
L minus 1.
So that's correct, L minus 1, and then don't forget that now there's also an E binding.
No, that is just for one.
Yeah, it's for one. You put it here. Ah, okay, great. Okay, so that's good. So now that's what comes in the denominator.
Yes.
Okay. So now in the numerator
This part.
you have that. Okay, excellent, good. So okay, so then the rest is algebra. Okay, so it's not so interesting. But so if you can, if with this assumption you can simplify things, okay? So I just say this, so for example, for your exam, you should be comfortable being able to do this type of a problem with a bit of a massage. So of course, I'm not going to give you exactly the same. I'll give you something else, but you know. So wait. We'll first start with. So you have these number of sets. Okay. You want to put these many in here. So the first thing you have to ask, how many ways are there to put these number of letters in this? That's the first question. No.
What is this E?
E solid is the interaction energy of each L, each of those spheres, with their environment. So let's start with the first question, which is when there's no ligand in the receptor. So I have L ligands in solution. So that so each of those has an interaction energy of E sol. Okay? But remember that there's not only one, there are many ways to permute. Okay. So you have to first show how many ways are there to do that. So if I have omega sites and I put L, I want to put L of them into those omega sites. How many different ways are there to put?
I think it is.
No,
This is permutation.
no. Okay.
Just right, the number of leaves.
Exactly. Think of your factorials. Omega
Permutation.
Be more specific. What do you mean by permutation? Yeah. Tell me what the permutations are.
I don't understand.
This is the partition function.
Oh yeah, partition.
So this is one part of the partition function. So this is when all of them are in solution, right? Then there's one where there's, so it should be a sum of this plus when the ligand is at the site, I get an e to the minus beta epsilon binding.
I thought it comes through here. Like, you have E, the sum altogether would be.
Well, okay, so, but remember that
They have different
the
energies.
different energies.
So we have
So,
to say-
yeah, the EL is, okay, so it looks like you've separated it out here. But you're still, okay, this itself doesn't have, so this is when one site is occupied, the receptor site.
Yeah.
So there must be another term here when the receptor site is not occupied.
Oh, I see.
So that's the total partition function. Then the probability will be some ratio of this to that. This is only one part of it.
How
Yeah.
many receptor sites do we have?
One. Only one.
This is
Yeah. Because it's wrong. That's why. Because it's wrong. This is combinatorics. I mean, so ask yourself the following. So forget about ligands and receptors. I have 30 boxes, and I want to put 10 marbles into 30 boxes.
how we do this.
How many? That's the question.
Can you explain the energy splitting down?
So, each ligand, when it's not in the site, has an interaction E sol. So, when all L ligands are in solution, the total energy is L times E sol. And then E bind is the interaction when in this case, the ball interacts with the receptor with E bind. Okay? And then I have L minus 1 in solution. So the total interaction L minus 1 times E sol. Okay, so and just sort of quickly go through this. So so those are the parameters. The weight of having the receptor occupied will be e to the minus beta E bind times summation of all the weights of the multiplicity times e to the minus beta L minus 1 E sol. Yeah.
I thought the L minus 1, the L is the number of ligand?
Yes.
I thought it was supposed to be summation. It was supposed to be summation of the E sol of the ligands minus 1.
Summation of the
Summation of all the ligands minus 1.
so this term here, the summation is what you get when you do the combinatorics. It's this. So when I have L minus 1 ligands in solution, I have to ask myself how many ways are there to put L minus 1 ligands in omega sites? How many ways are there to put L minus 1 ligands in omega sites? There's only one way to put the bound ligand into the receptor, because there's only one site to bind. So this has no multiplicity. It has no combinatorial effect, so it's just e to the minus beta E bind. There are L minus 1 ligands in solution, okay? The total energy associated with those L minus 1 ligands is L minus 1 times E sol. Yes? It has to be. Each ligand has E sol interaction. The L minus 1 of them, so L minus 1 times E sol. Total energy. So the Boltzmann weight, the Boltzmann probability, the probability associated with that must be e to the minus beta of that total energy. Okay. But there are many ways to have L minus 1 ligands in omega sites. So I have to do my combinatorical factor or the multiplicity here. Okay, so that's the weight of the occupied receptor. Okay. So that's the same as this thing on the right. If I want to know the probability, I need to know the partition function. The partition function is a sum of this thing along with all the weights of microstate 1, 2, and 3. I have to sum over all of them. And that is just the summation of e to the minus beta L E sol. Of course, I have to multiply by the multiplicity. Not clear.
I'm really confused.
Okay?
I mean, the ligands, all of them, do they have the same energy?
That's the assumption of the model.
They have the same energy.
That's what, when the ligand is in solution, when it's out, not in the receptor site, each of them interacts with the sol with E sol. That's the assumption.
Okay, so I think that the L minus 1 means that
You've taken away 1.
1.
Yeah.
Okay.
Exactly.
So if normally it was supposed to be e to the minus beta E, which is the energy of the system, then for this case, you say it is e to the minus beta L minus 1 times E sol.
Yes.
I was thinking it was supposed to be the sum of each energy per particle. So each of the energy of the particle was supposed to be the sum of ligands.
Wait. So, okay. Each of these, so let's, so it's not here. Okay, so we have this situation. Okay? Now there are L minus 1 of these guys outside. Okay. So the total energy in our simple model that contributes to the L minus 1 ligands being outside of the receptor site is this. Do we agree with that first or not? You don't agree with this.
So maybe I don't understand, that's why. So maybe I was thinking that instead we're supposed to add the energy of each particle.
You are adding the energy to each particle. This is
all the pictures have same energy. That's why you can.
It's E sol plus E sol plus E sol plus L minus 1 time. So I'm doing that here. So if you want, I mean, this is equal to.
So the heat that something is just a random of heat. Okay, that's right.
Okay. Okay. Good. Okay. So you're thinking of something more complicated when these will not be the same. Okay, but it is the same for me. So, okay. So now we agree with this. When I have this empty, now I have L of them. Okay, so I get L E sol. And now I have to multiply the Boltzmann factor, and I just do the same multiplicity, so the combinatorial part. Okay, so the probability of the ligand being occupying the receptor site is the ratio of this, to this thing. Okay, now that's the important part for my concern here. The rest is all about making some simplifications. Okay, so if the number of sites is much bigger than L, you can basically simplify each of these expressions of omega factorial over omega minus L factorial as omega to the power L. Okay, and if you want proof of this, you can. I mean, it's very easy to see. If you've got several orders of magnitude, omega sites bigger than L, you can easily show yourself that the leading order term in this type of an expansion is very well simplified by just saying it's, for example, 10 to the 6 to the power 5. Okay, so it dominates compared to the other terms. Only when omega is much bigger than L, which is the case for our model. Okay, so you do a bunch of simplifications, you write this ratio, and with a bit of math, you get something that looks like this. Okay, and this is what we want. This is what I want. I want to have an expression of the receptor site being bound in terms of an energy difference. An energy difference involving the binding energy and the energy of the ligand being in solution. Okay? Now, if we take this expression and compare it to what we had earlier on, so this is what you get from chemical thermodynamics. This is what you get from a simple statistical mechanical approach to a lattice model. And they look very similar. And what this allows you to do then is to write the dissociation constant. Remember, this is something that was related to an equilibrium constant of concentrations from an experiment in terms of something that we can think about as a physicist. We can think about energy differences between the ligand being in the solvent versus being bound to the receptor. Okay, so that's one of the reasons I really like this type of lattice model is it really helps you make a connection between the stat mech world and the world of the experimentalist who thinks of dissociation constants or equilibrium constants. Okay, so, well, there are many assumptions that I've made, or we've made in doing this. One of them you pointed out. Of course, not every ligand will have the same interaction energy in the solvent. What else? What else have I missed? I've also not included the ligand-ligand interactions. Okay, so that's something that could be put into the model. There's also the possibility that I don't have only one ligand. I might have ligands and other things. Different types of ligands. L1, L2, L3, L4. Okay. But the same type of arguments could be used to understand how, if I introduce other ligands into my lattice model, how does that change this thing? Because that's the type of thing that you should try to think about. Okay. Okay. Good. Are there questions on this before I move on? Yeah.
For these partition functions,
Yes.
should it be like omega minus 1? Because we are excluding the receptor.
No, that's not part of the lattice.
That's not on the part of that.
That receptor site is not part of the omega. It's the protein. But it won't matter. I mean,
We can start with.
you can start with omega, so yeah. Okay. Clear? Okay, so now we're going to switch gears a bit into building up on chemical equilibrium. Okay, so now you should be,
Curiosity, you asked about different color of ligands.
yeah,
Is it a legit problem?
is it a?
Like a legit problem, or just a kind of a mathematical curiosity in general?
No, it's a very much it's a relevant problem.
Because because in this case, one picture will have several other sub-pictures.
Yeah.
This picture will have several
So-
other sub-pictures.
So here's where it becomes relevant. In a cell, I don't have only L, I have a crowded environment. So if I have a crowded environment, how is that going to affect my probability of the receptor site being occupied by a ligand? So,
Because we have a similar problem in 1D where we
Yes.
have different color of bits.
yes,
Now in this picture we have 2D problem.
Yeah.
The same problem.
yeah,
And the problem becomes more complex if some of the ligands are same, some are of different colors, because there are other extra factors.
absolutely,
See,
absolutely.
so I hadn't thought about this 2D permutation problem. Now permutation becomes important.
Yeah, well, permutation becomes important, but then also the EL1, EL2, or ELI, ELJ interactions become important.
I think if we can find the number of things, it's just assigning the number, and like if we can categorize energy
Yes.
is just at the power. Like we have this number to the power this. If we can find the categorize the total number, that wouldn't be a problem. I think.
It wouldn't be a problem, but think of this. So right now, we're sort of also, well, okay, so if I have ELIJ interactions, and they become larger than E bind, then those interactions will compete for in a non-trivial way.
Okay, so we should also take what picture are possible, what picture are not possible.
Exactly. So it's not just a combinatorical issue then. Yeah.
So I just want to ask physically,
Yeah.
is there anything I mean, if you have different ligands, is there any special properties that makes the site attract?
Yes, many. So right now we've ignored. Actually, you should be able to answer this. So okay. So now, if I have charge, now there's no electrostatics here. Okay, I have charge here, and I have it's buried in here. Okay, but if you start thinking about how the charge couples with screening of the solid, then there are details that will matter. Okay. Okay, good. So now we're well calibrated on chemical equilibrium constants. Now we want to understand better equilibrium constants in liquids and in water in particular. So this is something that you learned in high school or in primary school. Okay, high school. In high school you learned that once every 10 hours, well maybe you didn't learn that, but once every 10 hours, a water molecule will break. It ionizes. And when it ionizes, it forms these two species. This equilibrium determines pH. So pH is minus log concentration of this. This equilibrium determines pH. And in your glass of water that you drink, there are the concentration of protons and hydroxide ions at pH equals 7 is 10 to the minus 7 molar. Okay, it's very small. So the concentration of water is 55 molar. So relative to 10 to the minus 7 molar. So there's a very low concentration of protons and hydroxide ions in neutral conditions. But when you go to acidic conditions, or when you go to basic conditions, the concentration of this or this increases. Now, this is something that we teach, well, people teach in chemistry, but actually it's such a basic fundamental process, but the actual mechanism or reaction coordinates by which this happens and how it changes as a function of pressure and temperature is not understood. Because it's a very complicated process, this ionization. If you can think of, it's a really nice problem in many body physics, many body chemistry, because it involves quantum phenomena, electron reorganization, zero-point energy, tunneling, all the interesting things happen in this process. Okay? Now, why do we care about this? We care about this because these guys here can affect the stability of protein structures in highly non-trivial ways. So this is something that happens in water. But if you go back to your peptide backbone, remember there's an NH group. Okay? The NH group, in the way that I presented it to you, it was like a static, rigid object. But actually, when you put an NH group in water, the proton here can exchange with the environment. So very rarely, but it happens, a proton will hop onto the solvent, it dissociates, and then it gets exchanged by another proton. Okay, so this is a very well known and established process in biology, and it's a way to study how exposed different amino acids are to water. So if you had an amino acid that was buried inside a protein, this would not happen because it's too far away. But if it's exposed to the water, you'll have this exchange. Okay?
Excuse me.
Yeah.
But it's not really proton, right? It's electron.
No, it's not a hydrogen atom that dissociates here. It's actually a proton.
The atom, I see.
Yeah. And I'll come back to your question because it's an important one. But this is a very basic process, and it's very similar to this. Okay? Here you take a water molecule, you split it. How much energy do I need to split the water? How much energy do I need to split a water molecule to break? Yeah, order of magnitude. 10 10 what?
Could be 10.
Are you sure?
I think so.
No. To break a covalent bond, how much energy do I
It's
need?
100.
Okay, good.
So if you have two covalent bonds,
Yeah,
two strokes,
yeah.
you go 2 times the magnitude.
To break two covalent bonds.
If you have to spend, if on one you have to spend 100.
Yeah, well, so here remember you're not breaking two covalent bonds.
Because you said you split.
Yeah, but again, here you're not splitting two. You're splitting one and then you're forming another one.
But, okay, yeah, I'm wondering, I mean, if you say you take water molecule and split, is it that the water molecule reacts with some Asian group that is around the surrounding? Because normally if you say water, you have 2 hydrogen.
Yeah,
So you have an atom. So if you look at the right of the equation, you have 3 hydrogen.
Yeah.
3 hydrogen,
Yeah.
H3, so
yeah.
beat my imagination. Where is the extra? Well, let's in the environment you have some H groups, OH groups, or something else.
Hold on to your question. It will become clearer shortly. Okay? Okay, so let's actually I'm not sure if this is actually true anymore, but I just show it to you anyway. So this guy, Dr. Warburg, who got the Nobel Prize in Medicine, made this claim that there's some correlation between development of cancer and acidity. So the notion is that cancer cells are more acidic in the environment. This has been a heavily debated topic, and well, this is just for fun, okay? I should have just removed the slide from the top, but anyway. Okay, so now, if I want to construct a model. I want to construct a model that simulates or models pH equals 7. Okay, so I have a box of waters. I have a box of water molecules, and I want to know how large does my water box need to be in order for there to be conditions of pH equals 7. So another way to ask this is the following. So if I take a volume of 20 angstrom cubed versus 50 angstrom cubed versus 100 angstrom cubed, what would be the pH if I put a proton in each of those conditions? Okay, so pH is minus log concentration of protons. So the number of protons per unit volume. Okay? When I go to very low pH, more acidic. When I go to very high pH, high OH minus, pH 7 equal. Concentration of proton hydroxide. So if I put a proton in a box of waters, how large does the box need to be before I get to a regime where the conditions, the concentration is equivalent to pH equals 7? So that's the question. And so if you remember, we asked a very similar question when we were talking about the concentration of proteins in cells. And then we said, let's put the proteins on a lattice. And then we said, what would be the typical distance between protein molecules at different concentrations? The higher concentration, the shorter the distance. So it's exactly the same question.
Are you putting just one proton?
Yeah.
But
No, it's not. Okay. What I'm saying is, of course, there's not just one proton. What we're saying is that if there are two protons, what would the distance need to be between them to resemble a certain concentration?
so is this just a model question? Or
Yeah, it's a model
general
question.
should be, if there are some high number of protons, what should be the average distance between two protons so that the pH is 7?
Well, you can't ask the question whether high or low, because there you assume that it's high or low. But what you can say is, so I fix pH equals 7, equal concentration of protons and hydroxide ions, and now I ask, what would be the typical distance between two protons? Okay, that's the question. So I fix pH equals 7, 10 to the minus 7 mole per unit volume. What would be the typical distance between the protons? That's the question. Okay. And this is just a figure that shows this to you. At pH 7, the typical distance between two protons is about 250 nanometers. 2,500 angstroms. They're very far away from each other. When you go to very low pH, that's when the distances become small. So you'd have to go to pH 1 so that the distance between the protons come to a length scale relevant to the Biram length. The Biram length being about 7 to 10 angstroms. So let me put this physically for you. In your glass of water that is at pH 7 or close to 7, 6.8, the distances between two protons is about 2,500 angstroms. They're very far away from each other. So they essentially, they don't feel each other, they don't talk to each other. Okay? But what this means is also, if you want to build a model to capture the conditions of pH equals 7, for the most part, you can neglect the protons. It doesn't matter. You can just have your simple lattice with just water molecules. You don't need to think of the protons in there. Is this clear or not? Because, you see, you could have asked me, in some variation of this model, one of the ligands could be a charge. It could be a proton. So the question of pH, so remember we discussed pH is a relevant variable that can change the stability of a protein. Okay, so if I change the pH, if I'm at pH 7, the number of protons within a distance of 2,000 angstroms is so small that it's not going to affect the thermodynamics or the dynamics much of the protein. Of course, if I start going to very low pH or very high pH, then it will matter, because at high pH, the distance between the hydroxide ions, the negative charge, becomes smaller. So there's an equivalent plot like this of POH. POH is minus log concentration of OH minus, and it will be something like that. It would be the mirror image, essentially, of this. Okay, so protons equals hydroxide neutral, acidic, more protons versus hydroxide, basic, more hydroxide versus protons. Okay? Now, I want to come back to your questions, so this will help you sort of understand what's going on. So, this is a movie of a proton and a hydroxide ion. Okay, this is the proton, is oxygen, hydrogen, hydrogen, or 3 protons. This is OH minus. Okay, and these are water molecules. And now you're going to see some interesting things happen. You just saw here, for example, that the proton, See, it forms a covalent bond, comes back, forms a covalent bond, comes back. And now something interesting is going to happen. Okay. This is, you're videoing this. Okay, so let's go back. I'm looking at this equilibrium, and I'm looking at the time reversed process only, because this direction is very slow. Why is it slow? Because I have to break covalent bonds. It costs a lot of energy. However, this process, once they come close to each other, is very fast, because there's a huge driving force for the system to go to this state here. Okay. And so what are the things that you observed here? So again, the anion and the cation, positive, negative charge. What I want you, so if you, let's imagine another type of cation, a potassium ion or a sodium ion. Does a potassium ion or a sodium ion move in the same way as a proton? Okay, let's wait till the end. Mm-hmm.
This last guy is what?
Which one? This one?
Yeah.
This is H3O plus. So you take a hydrogen atom, you pull out an electron, you create this guy in the gas phase. Now when you put this in water, there's some very complicated electronic structure and interactions that take place. So it does not become let's put the O here. It doesn't form anything like this, but what it forms is H3O plus. So essentially, the wave function of the proton is delocalized over several atoms. And so you have a species that looks like this. And that's what you saw here. And what you saw also, that's if you took a potassium ion, so a potassium ion has a very similar hydrodynamic radius to a hydronium or a this is known as a hydronium. These two have the same hydrodynamic radius. How would you estimate the diffusion constant of an ion from its hydrodynamic radius? What is it related to? Well, I've already given you the answer. Well, how is the diffusion constant related to the size of an ion? If I have an ion in a liquid with a certain viscosity and a certain size, the diffusion constant is given by its Debye-Stokes expression, which says that the diffusion constant is proportional to the radius divided by the viscosity. The higher the viscosity, the slower the diffusion. more friction. The larger the radius, the slower the diffusion. Okay, so, okay, these two have the same hydrodynamic radius, but the diffusion constant of this is much larger than this. So Debye-Stokes theory breaks down. And you saw why it broke down, actually. It broke down because, you see, at the end, you saw that many protons moved at the same time. Okay, there was a, let's go right there, right there, right there. Bring it to, see? Let's play from here. See, they all simultaneously move together. This is very different from the way a standard ion moves through water. When a regular ion moves through water, it drags the solvation environment with it. With the proton, there's covalent bonds being formed and broken. This is why it's a very interesting but also very hard problem. Okay, so Joel, I hope this sort of answers what you were asking of what. Okay. Okay. Good. So now, with that in mind, we're ready to discuss acid-base chemistry in amino acids. So, equilibrium, I have an acid, and there's a general acid, and when I put it in water, it dissociates into a proton and a general base. So, you all know this. I put HCl in water. Hydrochloric acid is a strong acid. It dissociates into proton and chloride ion. That's why it's a strong acid. Okay, so this is an example. This is acetic acid. Acetic acid, in this case, has the total charge is 0. When I put it in water, it can dissociate, and when it dissociates, it loses a proton, and so the total charge of the system is -1 now. So the reason why we emphasize a lot on acid-base equilibrium is because it changes the electrostatics a lot, and this, you should know by now, will affect the stability of your protein. Okay, so now in the same way that we wrote the dissociation constants for the ligand binding problem, the dissociation constant is essentially a ratio of the concentration of the product of these two guys to this. The pK, okay, is like pH, is minus log of the concentration of this, or the ratio of the concentration. So it's, again, a measure of the tendency of the molecule to dissociate. That's all this is. It just puts it on a scale that allows, a log scale that allows us to understand it or see it more clearly. Okay, so you all probably saw this at some point, but I just remind you. So this is the dissociation constant. pK is minus log of this. If I write it out, I get this thing known as the Henderson-Hasselbalch equation. So it tells me a relationship between pH minus log of the concentration of protons, the dissociation constant of my molecule or my substance, plus the log of the ratio of these, the general acid and the base. Okay, now with this equation or relationship, I can determine when I put an amino acid in water, and if I know the pH, and I know the pKa, which we will discuss shortly, then I can tell you what is the equilibrium state of the amino acid in water. So I can tell you whether the system wants to be in the protonated form or in this form. Intuitively, you already know. When I go to very low pH, this will dominate. When I go to very high pH, this will dominate. But at what pH will it change? Okay, so if I told you at pH 3 or pH 4, what's the protonation state? We need to have a way to be able to determine that. And this expression here allows us to do that. Okay, because if I know this, and I know this is something that is known, for example, experimentally, it's given to us, then I need to measure or know what the ratio of these two concentrations are. If it's greater than 1 or less than 1, it tells me which is the dominant species in solution. So we're going to do some exercises together to make sure we understand this. Okay, so every group, every titratable group, a titratable group is a group like the following. So it can exchange protons with the environment. Every titratable group has exchangeable protons. Those sites can change their protonation state and their charge. CH groups, are they titratable? Can a CH group exchange protons with the environment? It could, but it would cost a lot of energy. It'd be very difficult to do. Okay, so different sites have different tendencies to exchange protons. That thermodynamic driving force to exchange protons is given by the pKa. So it's known as the pKa. Okay, so pk something is always minus log of some equilibrium or dissociation constant. Okay? Okay, so not all amino acids have exchangeable protonation sites. So these are examples. This is an example of lysine. It has an exchangeable protonation site. But let's take, well, I don't have the example here. Let's take, if we take, this is glycine. Glycine, this is its R group, the side chain. It can't exchange protons. Okay? Alanine. Alanine is CH3. This is the R group. It can't exchange protons. But if the side chain has a group like an NH3, NH2, or a OH, or a O minus, okay, so a CO minus group, then it can exchange protons with the environment. And so we wanted to be able to know and to understand, if I give you an amino acid or a protein, and I want to tell, and I say I'm working at pH 1, pH 7, or pH 14, what is the total charge of my protein or my amino acid? That's the kind of question that we wanted to be able to answer. Okay? Okay, so don't forget that every amino acid has the N and the C, the two N's. And the two N's are made up of groups that can also exchange protons. Okay? Okay, so I'm going to give you, probably we won't finish it today, but this is yeah. So typically it's nitrogen and oxygen. They're the elements that can exchange protons. And the charge is coming from the fact that those elements are either losing or gaining protons. That's where the charge is coming from. Okay, so Okay, so this is the question. Okay, so I think we're going to continue tomorrow anyway, but I just pose the question and then we'll continue. So this is the relationship that we have. pH, pK, and this log of two terms. I want you to start with lysine, and I want you to tell me if I take lysine and I take it at pH 5, what is the total charge on lysine? And if I take it to pH 12, what is the total charge on lysine? That's the question. And you have all the information to be able to answer this in the two documents that I gave you. Now, you'll see that in this one here, there are 3 columns. pKa of the C terminus, pKa of the N terminus, and pKa of the side chain. So all amino acids have the C and the N. Because all amino acids have the C and the N terminus. Yeah? This guy. The blue circle and the red circle. But if you look at the side chain, not all of them have it. Because not all amino acids have titratable side chains. So for example here, this is glycine and this is, I think, isoleucine. If we look at glycine, there's a blank value, isoleucine, also blank value. So the total charge in this system would only come from the N and the C terminus. Okay. Now I'm asking you, take lysine, and this is a single lysine in water. What is the total charge of lysine at pH equals 5 and pH equals 12? And I've just said this is the lysine has a pK of 10.53, but I don't know if this is the same here. It's okay, it's 10.79. You can use the one in this. It's depending on different measurements, different experimental measurements will give you different pKa. But so use the value in the chart that I gave you. So the only tricky part of this that you need to think about a bit is figuring out this. That's the only tricky part. So just think of it. Okay?
In that case, how did you come up with glycine, like take pKa as in 10?
Okay. So that is measured experimentally. It can also be something that can be computed theoretically. Essentially, it's related to what is the reversible work required to take lysine and put a proton on it or take away a proton. That's what it's related to. It's related to a free energy, which can be measured.
So,
Yeah.
please, can you go to the general relation of H and
Okay, sure. This one. Maybe, actually. Let's see. Probably, yeah, this one.